Nucleic acid hybridizations. A presentation from the Zoology Department GDC wrapper. Random prime DNA labeling. The random prime DNA labeling method uses a mixture of many different hexanucleotides to bind randomly hexanucleotide sequences in a DNA template and prime new DNA strands synthesis. Synthesis of new complementary DNA strands is catalyzed by the Col-Now subunit of E. coli DNA polymerase I, containing the polymerase activity but not the associated 5-3 exonuclease activity. RNA probes are made by transcribing cloned DNA. The plasmid vector PSP64 contains a promoter sequence for phage sp6 RNA polymerase linked to the multiple cloning site, MCS, with an origin of replication, ORI, and ampicillin resistance gene, AMPR. A suitable DNA fragment is cloned into the MCS and recombinant DNA is opened by cutting with the restriction enzyme VUL. sp6 RNA polymerase and a mixture of NTPs, at least one of which is labeled, are added, initiating transcription from a specific site within the sp6 promoter. Multiple labeled RNA transcripts of the inserted DNA are produced. Fluorophores are used in non-isotopic labeling. Nucleic acids can be labeled with chemical groups that are not detected directly, R. The incorporated groups are bound with high specificity by an affinity molecule, A, carrying a detectable marker, M. The marker which carries a fluorescent dye, Mar complex, which is detected by fluorescence microscopy. Blot hybridization offers rapid screening. Blot hybridization offers rapid screening and often employs allele-specific oligonucleotide probes. In dot blotting, the test sample is an aqueous solution of purified DNA or RNA, for example total human genomic DNA. The variant technique of slot blotting involves pipetting the DNA through an individual slot in a suitable template. In both techniques the DNA are denatured before hybridization with a denatured labeled probe. Allele-specific oligonucleotide, ASO, probes are constructed from sequences spanning the variant nucleotide site. Hybridization in which DNA duplexes are formed between probe and target. The duplexes formed are only stable if there is no mismatch. Hence the technique can be employed to detect even a single pair mismatch as in sickle cell disease. Sickle cell mutation testing. The sickle cell mutation is a single nucleotide substitution, AT, at codon 6 in the beta-globin gene, resulting in a GAG, GLU, GIG, VAL, substitution. Allele-specific oligonucleotides, ASOs, were designed to match the nucleotide sequence flanking the substitution. The schematic dot blots show the results of probing with these two ASOs under hybridization conditions. In situ hybridization. Some hybridization assays rely on hybridization to target nucleic acid within fixed chromosomes or cells trapped on microscope slides. Because the nucleic acid is immobilized within native structures, the hybridization is said to occur in situ. Chromosome in situ hybridization is a simple procedure in which microscope slide chromosome preparation is made, using metaphase or prometaphase chromosomes. RNA and protein are then removed by treatment with RNase and proteinase K, and the remaining chromosomal DNA is denatured by exposure to formamide. The denatured DNA is then available for in situ hybridization with a labeled nucleic acid probe, overlaid with a cover slip. Tissue in situ hybridization. Very thin tissue sections are cut with a cryostat either from tissue blocks embedded in paraffin wax or from frozen tissue, and then mounted on glass slides. A hybridization mix including the probe is applied to the section on the slide and covered with a glass cover slip. Tissue in situ hybridization. The example shows the expression pattern produced by hybridizing a digoxygenin-labeled sonic hedgehog antisense riboprobe against a section of tissue from a mouse embryo at 13.5 embryonic days. The section is at the level of a part of the forebrain, top and middle, and the upper and lower jaws, bottom. Detection of the probe was by an alkaline phosphatase-coupled blue color assay. Strong expression was seen in very specific region S of the developing central nervous system, as indicated, and also in the secondary palate. Colony blot hybridization. When a nitrocellulose filter is placed on top of bacterial colonies on an agar plate, cells from the colonies adhere to the membrane. The membrane is transferred to a new culture dish to grow the cells into colonies. The replica membrane is then hybridized with a labeled nucleic acid probe. After a wash, X-ray film is used to create an autoradiograph showing the position of labeled colonies on the replica membrane. The positive colonies can be picked and grown individually in liquid culture. Principle of microarray hybridization. A microarray is a solid surface on which molecules can be fixed at specific coordinates in a high-density grid format. Microarray hybridization uses oligonucleotide or DNA microarrays that are prepared by fixing thousands of synthetic single-stranded oligonucleotide or DNA probes at specific predetermined positions in the grid. Each specific position will have many thousands of identical copies of a single type of oligonucleotide or DNA probe. A test sample containing a heterogeneous collection of labeled DNA fragments or RNA transcripts is added to hybridize with the probes on the array. The target sequences might result in a strong hybridization signal for some. For other probes there may be few target sequences, resulting in a weak hybridization signal. After washing and drying, the hybridization signals for the thousands of different probes in the array are detected by laser scanning, giving huge amounts of data from a single experiment. Microarray hybridization. Individual oligonucleotides or DNA are fixed on each coordinate on grid. Test sample of nucleic acid with fluorescent label are added. Various target sequences are formed. Laser scanning is done to detect a strong hybridization signal. In addition to microarray hybridization, microarrays have many other applications like protein arrays, arrays that assay binding between nucleic acids and proteins, and many others. References: Biotechnology, 2nd edition. David P. Clark and Nanette J. Pazdernik. Academic Cell. Medical Genetics, 3rd edition. Dorian J. Pritchard and Bruce R. Korf. Wiley Blackwell. Human Genetics and Genomics, 4th edition. Bruce R. Korf and Myra B. Irons. Wiley Blackwell. A Zoology Department GDC Raper presentation. Thank you.